Hello, IB Bio students. Today we continue with 11.4. This is the last section of topic 11, and we're going to look at gametogenesis. And we are starting with the initial understanding that spermatogenesis and oogenesis involves three things. It involves mitosis with cell growth, it involves two meiotic divisions, and it involves differentiation. So when we look at gametogenesis as a whole, we can divide that into spermatogenesis for males, and this is where sperm is produced. The correct or full name for sperm is spermatozoa. In females, the process is oogenesis, and this is where eggs are produced, and the scientific name is the ova. And we know that all of this gametogenesis happens in the gonads, which are the organs. So ovaries for females and testes for males. So here we see the processes that are three processes that are involved in this gametogenesis. So spermatogenesis is a lot more simple than oogenesis. So spermatogenesis takes place in the seminiferous tubules of the testes. Now the seminiferous tubules, we're going to look at the structure in the next video, but they're lined by this germline epithelium. So the process of spermatogenesis begins at puberty. And this is where this germline epithelium begins to divide by mitosis. So these cells here begin the process of mitosis. And these cells, or the new cells that are formed, are called spermatogonia. And notice that these cells are diploid. So these cells undergo a period of mitosis and growth, and they turn into primary spermatocytes. And then the primary spermatocyte undergoes meiosis 1, and it turns into secondary spermatocytes. So here we have two haploid cells now, because the first meiotic division has happened. Then the second meiotic division happens, and you end up with 4 haploid cells or 4 spermatids. Now, these spermatids still need to differentiate to become functional sperm or spermatozoa. So pretty simple. The process of oogenesis is a little bit more complicated. This happens in the ovaries, to a lesser extent in the oviduct, because the final phase of ova production happens when the sperm fertilizes the egg, and that happens in the oviduct. So one key difference between spermatogenesis and oogenesis is that the process of oogenesis begins during fetal development. So here you start with a whole bunch of primordial cells or germ cell lines that are formed by mitosis. So it gives you approximately 40,000 primordial cells that are eventually going to turn into eggs. So these cells, or oogonias, undergo cell growth until they're large enough to undergo meiosis. So here you see the oogonia. It is diploid. It's lined with these granulosa cells for nourishment. And then during that prenatal stage, fetal development, they just grow, right? They undergo mitosis and they just grow. And they turn into these primary oocytes, which begin meiosis but do not finish it. So they get locked in prophase 1 of meiosis 1. Okay? So notice that the primary oocytes here, they are surrounded by these granulosa cells, and they are now called a follicle. Okay, so because they're surrounded by these cells that provide nourishment for them. So the primary oocyte surrounded by the follicle, surrounded by these cells to form a follicle. So here we see the cell that is locked in prophase 1 until puberty. And during puberty, the girl begins her menstrual cycle, which is all hormone regulated. So follicle stimulating hormone is going to trigger the menstrual cycle, and is going to trigger the release of this primary oocyte, which will continue to divide via meiosis 1 into a secondary oocytes. Okay? Now, what happens when the secondary oocyte right here forms. So the secondary oocyte is diploid, but also something known as a polar body forms at the end of meiosis 1. And this polar body, notice that it's a lot smaller, it's unequal in size, it has less cytoplasm. And notice how this polar body is just kind of trapped in here inside this follicle until it could A, degenerate, or B, differentiate into another polar body. Okay, so the secondary oocyte gets locked in metaphase 2 of meiosis 2. So it hasn't quite finished meiosis, so it's still inside the ovary. So once ovulation happens of this particular egg, because remember, not all eggs are released at once from the ovary, but when this egg gets released and ovulation therefore happens, so ovulation happens here, ovulation, I can't spell. What happens? Well, the continuation of meiosis 2 continues when and if this secondary oocyte gets fertilized by a sperm. So ovulation means that this secondary oocyte is released into the oviduct, right, or the fallopian tubes. And it might or it might not get fertilized by the sperm. If it does get fertilized by a sperm, then this secondary oocyte will complete meiosis 2. So here we'll see the finish or end of meiosis 2, finally when the sperm fertilizes this egg. What does that mean? That means that there might be another production of a polar body here. And so now you might end up with 3 polar bodies and the ovum. And the ovum is the mature egg. So this is the egg that is now ready to fuse, or the nucleus of this egg is now ready to fuse with the nucleus of the sperm and then form a zygote. So here's a nice video that shows the formation of the polar bodies. Here you see it in a time-lapse video. There's the polar body here forming off to the side. Okay. And there it is. All right, so there are some key differences between spermatogenesis and oogenesis, and this is what's going to lead to a different number of gametes with different amounts of cytoplasms. So what are some of the key differences? 1, the number of cells produced. 2, the size of the cells produced. And 3, the timing of the process. So here you see the summary with the key differences. So let's start with the process itself. As far as location, spermatogenesis happens entirely in the testes, oogenesis mostly in the ovaries, and since the end of meiosis 2 happens after fertilization of the sperm, the rest continues in the oviduct or the fallopian tube. As far as meiotic divisions, you have equal divisions of cells, which leads to 4 haploid cells, whereas oogenesis, you have unequal division of cytoplasm, which leads to the polar bodies. In spermatogenesis, you have the germline epithelium involved in gamete production, where in oogenesis, you do not. You produce 4 gamete cells in spermatogenesis and only 1 functional ovum in oogenesis, and then anywhere from 2 to 3 polar bodies. As far as the size of the gametes, the sperm is smaller than the spermatocytes. In oogenesis, the ova, or that functional ovum or egg, is larger than oocytes. The timing, spermatogenesis is uninterrupted, and it begins at puberty, and it's continuous. Whereas the timing of oogenesis is in arrested stages. We saw that it begins in the prenatal stage, and then we saw it arrested again, and then it continues again after puberty. And then we see that meiosis 2 doesn't happen until fertilization of that egg. Spermatogenesis is lifelong, but it reduces with age, whereas oogenesis terminates at the onset of menopause of a woman, which depends on the age of the woman, sometime around 45 to 55 years of age. So here you have the gametogenesis comparison. Oogenesis, you start with the primary oocyte and you end up with one haploid functional ovum. In spermatogenesis, you start with a primary spermatocyte and you end up with 4 functional spermatids, which will then differentiate into a functional sperm. Okay, that's it for the first section of 11.4.